go ahead and start off. So the first thing is I forgot to give you guys your study tip last week, but I remembered for this week. So it's called the bridging game. So whenever you have a chapter or even just a lab like for us, you can write out all the different terms and concepts, just random pieces of paper, however you want to do it. Just make sure it's just one term or one concept per piece of paper. Throw all those pieces of paper into like a box or a large hat, a large bowl, whatever. And then at random, pick two at random and see if you can explain how they're related to each other. And the explanation is the bridge. So building a bridge of ideas between the two terms. How could they be related? What does one have to do with the other? And that will really boost your understanding and your memory. And also, as you do it, it'll help you recognize if you're struggling, you're going to realize, oh, I need to study those concepts in particular a little bit more. Okay, so enough about that. This week, you're going to watch a TED Talk, which is presented by the co-inventor of the CRISPR-Cas9 system. It's about 14 minutes long. It's short. So here's a link, but also in the discussion prompt, you have a link. And then I also just put the link in with your simulation at the very beginning of your simulation as well. So this link is everywhere for you. As you listen to her presentation, you can pause and take notes. And then you'll see that the first 4 questions on your assignment, discussion assignment, are based on that talk. And then the last question will be based on the simulation. So then you'll have the eutrophication Labster simulation and the eutrophication post-lab questions as usual. So for this CRISPR, we just added one extra thing for you to do is watch the TED Talk, because it will really help you to understand the CRISPR system, in addition to my presentation that I'm giving you now. I imagine that the CRISPR-Cas9 gene editing system, along with the TGF beta and EMT scenario, is probably the most complex of all the labs we'll be doing. So yeah, want to make sure that you really understand it. Okay, so what is CRISPR? And this E should not be there. CRISPR does not have the letter E. I included another link here for you to see some good simple animations, which will be saying the same information that I'm going to tell you, but being able to see it more visually is very useful. So I highly encourage you to watch this YouTube video. And it's also short. Okay, so what is the CRISPR-Cas9 system? It's a bacterial immune system. Now, it's not like you and me. We make antibodies, and we also have a whole set of specialized cells to fight infection. Bacteria are single-celled organisms, so they don't have anything elaborate like you and I have for our immune system, but they do have this molecular system. It's actually a protein and RNA system that can seek out and chop up viral DNA. Okay, so CRISPR is an acronym, and see this, it's spelled correctly here. CRISPR, CRISPR, just no E. Okay, and so CRISPR, if you look at the bacterial genome, you'll find an area that's a CRISPR area. And what it stands for is clustered, regularly interspaced short palindromic repeats. And I know that's a mouthful and very overwhelming, but basically it just means you'll have these repeated sequences and they're not back to back. There's little gaps between them. And these palindromic repeats, palindrome means it's the same forwards and backwards. So you have these short, about 20 to 40 base pairs in length palindromes that are repeated, but there's also these short little spaces in between. And those in between spaces are called the spacer DNA. And that is where you get the unique short sequence. And that unique short sequence in between these palindromic repeats, these short unique sequences are the viral sequences. So this is how the bacterial cell has like a memory of viral infections. Any viruses that have come into it get put in between the short palindromic repeats so that it can, in a sense, have a memory and an immunity to it. Okay, so that's the first part. Then what we found is that a little bit upstream of these CRISPR areas are actually a related set of genes called the Cas genes because it's CRISPR associated genes, and in particular, the CRISPR associated genes code for the protein that becomes the active agent to destroy the virus. So the two genes in particular, the two Cas genes that we're particularly interested in, are helicase and nuclease. Helicase is going to unravel or unzip the DNA, and the nuclease is going to cut target DNA. But it doesn't do this randomly. It does this highly specifically. And so the Cas9 protein system takes the helicase and the nuclease, making it into this large protein complex, but then it also takes in RNA, and we call this single guide or gRNA for guide RNA. And this guide RNA is where you get the specificity. So if the G guide RNA is not able to bind specifically to the DNA, then you will not cut. So it's highly specific. And this guide RNA is actually composed of two pieces of RNA. One that is the specific target sequence, and that target sequence comes from that spacer, right, the unique part. And then it's also linked to another piece of RNA called the tracer RNA. The tracer RNA actually attaches to the protein, the Cas9 protein. And so then you get this complex, which you can call a, what's the word I'm looking for? Ribonucleoprotein. So ribonucleoprotein, ribo for the two pieces of RNA, the tracer and the target, which together can just be called the guide RNA. So you have the ribo for the RNA component, and then you have the helicase and nuclease part would be the protein part. Okay, so turns out that you can do a couple of things. You can use this Cas9 complex to simply specifically find and bind the target and make a cut. And it turns out that it will cut both pieces of the DNA. And that's really important. If you only cut one if you were to only cut one of the DNA strands, the repair system would fix it perfectly and there would not be any problem and nothing would result. But because we're making a double-stranded DNA break, now the only way that the cell can repair that is through a messy repair system called non-homologous end joining. So non-homologous end joining is just going to force those ends together any way it can. It's going to throw in random base pair or even remove another base pair from what's already present. Anything it can to get those ends to be able to be sealed back together. Remember, in order for the strands to fit together and keep the helix, right, you can't just put anything together. But by randomly putting in base pairs simply just to fit, you're definitely not going to maintain the original sequence. And that gene, therefore, will have a mutation. And that mutation will make the gene non-functional. Anywhere along the gene sequence, if you randomly play around with the sequence, you've changed what it's coding for. The amino acids that it codes for are changed, and then the protein can't fold correctly. It's non-functional. And so that is what we mean by knockout. You've made that gene no longer functional. You don't get a functional product. So you've essentially knocked it out. But we didn't remove the entire gene, right? We just made 2 cuts on both sides, right? Double-stranded cut. And then the repair system, the non-homologous end joining system, had to figure out a way to put the strands back together. And by doing that, it introduces a mutation, and the mutation renders the gene unusable, non-functional. And so then it's as if it didn't have the gene at all. Okay. So that's the really most important concept to understand. Okay. Now we can also, there are other methods that can be used to actually insert a gene. You would have to, in addition to introducing this whole system, the CRISPR Cas9 system, you would also need to introduce the donor DNA. And it's a little more work, but we can do it, and we have done it in animal models. And so both of these strategies, to knock out a gene or to add a gene, are both really useful in research, but also has huge applications, potential therapeutic applications to treat genetic diseases. So if someone has a genetic disease that's caused by a gene that normally would be silenced, or wouldn't normally be present at all, you could knock it out. If someone has a genetic disease because the form of the gene they have is not acting properly, then you could insert the correct version of the gene and cure their genetic disease that way. Huge potential, but you can't just start playing around with people's DNA. So this is not yet available as a therapeutic treatment, but it will be soon. And the video will talk about this. Also, of course, there's huge concerns about people editing genes in embryos and having, so-called designer babies. So instead of just using this tool to treat, cure diseases, any gene that you have the sequence for, and you know exactly where, how to design the experiment, you could potentially start just making like we said, the designer babies, things that people find desirable, right? So you could anything from height to, eye color. Now, we actually don't know the full scale and scope of how to do that yet. But again, the system has that potential. And so then we have all these ethical concerns of, well, should we start allowing clinical application? How do we control and prevent non-ethical uses, right? So there's some more work to be done in that regard. So we're not using this in medicine yet, but soon. Okay, so here is the person who gives the TED Talk keeps it real simple to a general audience who don't necessarily have any science background. This is a lecture by the same person, co-inventor of the Cas9 CRISPR system. And so it'll go into the detail that you need to know for the lab, as well as, of course, going through the Labster, reading those theory tabs and answering their questions and performing the experiments will also walk you through. But if you go into the Labster already having an idea of how it works, it'll be much easier for you to understand what's going on in the simulation, how you're utilizing the tools to solve other questions. Okay, so the next thing that you need to be introduced to is the scenario that the Labster simulation is, saying that you are doing research on EMT. So EMT stands for epithelial mesenchymal transition. This is where epithelial cells, which normally look like this, very regularly shaped, tightly adhered to one another through cell-to-cell junctions, right? And they have a regular basal matrix that looks like this. The cells have their shape, as I mentioned. And so this is the normal epithelial phenotype with the tight junctions, the basal membrane, and the cuboidal shaped cells. Then you have, oops, lost my picture. Then you have, under certain conditions, these cells can be transformed to become like mesenchymal cells, which changes the shape. It breaks those cell-to-cell junctions. The matrix is different. You notice that. And so you have a completely different phenotype in these cells. Now, this is a normal process when it comes to things like organ development, wound healing, right? Or even in the embryo during embryonic development. But this also occurs when it shouldn't occur. This is the same process that leads to tumor formation and even cancer. So we do want this when it's supposed to happen in these scenarios, like the embryonic development and wound healing, but we don't want this otherwise. When this happens otherwise, then it's going to be bad. It's going to be a tumor and maybe even cancer. Okay, now when we're talking about proteins that we find in the epithelial cells, you have things like E-cadherin, and that is a really important protein in these cell-to-cell junctions. Then you also have F-actin, which is a filamentous form of actin. You know of actin probably from muscles, right? But it's also found in many other types of cells, and particularly in epithelial cells, it's found predominantly around the edges. And this makes sense. These cells need to have that rigid cuboidal shape. So having the F-actin as a vital part of the cytoskeleton to give it its shape. Okay, so when they transform into mesenchymal like cells, you get the loss of the adhesion, right? You don't have those adhesions anymore. They're now free to move away. You've changed their shape. You've changed their cytoskeleton. You've also changed their extracellular matrix, and you've increased their motility. And so now they're able to break away and move around. Okay. And that's how you can easily see how that would be something that cancer, right? We know that metastatic cancer is cancer cells that break away from the tumor and move around throughout the body and start forming cancerous tumors throughout the body, right? So we know this is an important mechanism to study in the treatment of cancer. Okay. So then how are these cells being transformed into mesenchymal cells? And so one of the ways, so there's actually several ways that this can occur, but one of the ways is through a particular growth factor cytokine called transforming growth factor, and we abbreviate that TGF beta. Sometimes you will see it abbreviated further as just TGF-B, as you see here and here. Okay, so sometimes the beta is not spelled out and it's just abbreviated with the B. So when TGF-beta, the cytokine TGF-beta, binds to the cell membrane at its specific receptor, the TGF-B receptor, that is going to trigger a cascade of events that they abbreviate here with, it's not just 1-2 steps. It's going to be several steps. But eventually what happens is you get this complex of these SMAD proteins, which you can see here, a complex. And that SMAD complex is able to move from the cytoplasm into the nucleus and then act as a transcription factor. So the SMAD protein complex acts as a transcription factor. It's going to turn on or turn off particular genes. And that is how the epithelial cell gets transformed into a mesenchymal-like cell. Okay, so once you've studied in the Labster simulation, once you've gone over this EMT induction by TGF-beta, then it's going to have you take a cell line and expose it to the TGF-beta, and then you're going to do an immunofluorescence. It's just abbreviated immunostaining, but it's going to be fluorescence microscopy, and you're going to compare cells before and after being treated with the TGF-beta. And specifically, you're going to look at those two proteins I mentioned, the E-cadherin and the F-actin. So you're going to see what do these two proteins normally look like in these normal cells, and then what does it look like after these cells have been treated with TGF-beta? How have the two proteins changed? Have they changed? Do you have more or less? Are they in different positions? What has changed? And then once you have an idea, then you're going to make your hypothesis. You're going to then perform experiments to answer that. And so you will end with designing the experiments to utilize the CRISPR Cas9 knockout system. And so they'll have a section where they teach you about CRISPR before you go into designing and doing the experiment. So this Labster simulation is long, and I do recommend that you you take some breaks, just leave it up and running, and maybe go for a short walk, take a snack break, something, so that you can really take in all the information, take all your screenshots, take all your notes. It's a long one. Okay, so you will design experiments utilizing the CRISPR-Cas9 knockout target gene involved in the EMT phenomenon. Okay, so once the CRISPR experiment is completed, you would normally confirm that the selected gene was actually correctly knocked out. And so the steps to do that would be making positive clones, so selecting for the cells that you actually got the vector into. So in order to get CRISPR Cas9 system into cells that don't have them normally, so you're going from, bacteria, which have the CRISPR Cas9 system naturally, and now you're trying to utilize that model in like a mammalian cell, which would not have this system at all. And so to get that system into these cells, the cell line that you're working with, you have to use a vector. And of course, for us, that vector is going to be a plasmid, which you already know what a plasmid is. And so you will do electroporation to get that plasmid into your cell line. But something you may not know yet is that not every single cell in your electroporation experiment, not every single cell is going to take up a plasmid. And so you need to grow them. You need to select for the ones that actually took up the plasmid and have the CRISPR-Cas9 knockout. So you don't want cells that have the gene. You only want the cells that have the knockout. You want to select for the knockout cells. And so usually that would take a couple of weeks to do all of that work to get all of your positive clones, or what we call a stable cell line of only positive clones, the knockout cells. The simulation will actually do that for you. Okay. And then the next step would be to then take that stable cell line of the positive the knockout cells and perform PCR to amplify the target DNA. And then this is where it gets a little tricky, is that you are selecting for the gene that you're hoping that you knocked out. Now, remember, knockout does not mean you're getting rid of the entire gene. It just means you've made a mutation that renders it useless. So you're going to amplify that target gene, and then you're going to perform. You won't actually perform, but this is what you would do. The next step would be to take that amplified target and sequence it. And when you sequence it, you would look to see if it does indeed have a mutation. And then you would, of course, have to repeat this to confirm that it worked. You would also look for changes in expression or phenotype. And that's when you can do things like FACS cell sorting. You could do things like the Western blot, or immunofluorescence, which is what we'll be doing. So the simulation is going to do all this for you and let you do the immunofluorescence. And then once you compare the immunofluorescence results, then your part will be done, and that's pretty much the end of the simulation. So now you've had a very thorough overview of the simulation, what to expect. And you've had an introduction to what is CRISPR-Cas9 system, and what is EMT, and one of the ways that it's induced is through the TGF beta. Okay, so I know that's a lot of information I just threw at you. So by the time you finish watching these YouTube videos and you finish your simulation, you may be wondering, wow, that was a lot. What was I supposed to take away? So this slide here is to help you when you review all your notes. This will help you to know what exactly to focus on. So you should be able to explain what does EMT stand for, but also what does it mean? Explain it. Okay. What is the function of TGF beta? So it is a cytokine, but what does it do? So remember that binding to the cell receptor causes the cascade to lead to the transcription factor, right? Okay, so that whole system of how the epithelial cells get transformed into mesenchymal cells using TGF beta. Okay, how does that relate to cancer? Well, if you're turning on or off genes that allow the cell to become mesenchymal-like, you're allowing them to become cancerous, essentially, right? So if we can figure out a way to stop this transition in a targeted area, then we could perhaps stop the cancer, right? You should be able to explain to me what is F-actin and E-cadherin proteins. How are the expressions of these proteins affected by TGF beta? So when you do that comparison of before and after application of TGF beta, right? So make sure you take notes on that. So how does TGF beta application to the cell line change the protein expression? Does it increase or decrease? And make sure you know for each one of these. And then you'll need to know the details of CRISPR and the Cas9 system. So how is the CRISPR-Cas9 system used to knock out a gene? How is it specific? All of the components of CRISPR. So I didn't go over every single thing with you just now. There will be some additional information on the CRISPR-Cas9 system in the simulation. It just wouldn't have made sense to go over every little thing with you now, but just to give you enough introduction for it to make sense while you do the simulation. Okay,